Everyone, my name is Simon Villegas. I am a PhD student at the One Health Genomic Lab at the Universidad Nacional de Colombia, and today I'm going to talk about something that relates to every one of us, and it's going to make sense in a while what I'm about to say. I always like to start my talks with a question that it doesn't have seemingly nothing to do with the theme that gathers us here. So I'm been into mindfulness and yoga recently, and I would like to invite you to have just one conscious breath. We are not conscious how many times we breathe. Please meet Juan Restrepo, and he is teaching us how to breathe. After we have our breath completed, I would like to ask, what are you breathing? The obvious answer is air, oxygen, nitrogen, several gases, but that's not the whole picture, and that's what I'm talking about today. To give you an idea of my theme, according to the latest numbers from the health organization, the World Health Organization, more of 90% of the people in the global population does not breathe a good quality air. Even though there are several policies around the air quality in different cities. The bad air quality, it's accountable for around 4.2 million deaths per year. To give you an image of how big that number is, the whole population of Medellin, it's 2.5 million. So it's almost if Medellin dies twice in a year due to bad air pollution. In the case of Colombia, the air is the is the primary cause of the environmental borne disease, even more than water and other causes. And it causes several diseases. For instance, coronary artery disease, EPOC in Spanish, lower respiratory tract disease, and several infections. This is a problem not also from the public health perspective, but also from the economical perspective. To give you some numbers, in Latin America and the Caribbean, the costs associated with bad air quality are around between 1.5% and 3% of the gross domestic product. In Colombia, it's around 2%. And here, in the area metropolitana del Valle de Aburrá, which Medellin is located, it accounts for around 5% of the gross domestic product. So it's also an economical problem. The area metropolitana de Valle de Aburrá has some special topographic and meteorological conditions that hinders the air pollution, the contaminants in the atmosphere. As you can see in this picture, it's a deep valley. It has around 7 kilometers wide, and we experience several times a year a phenomenon called thermal inversion. What this means is that the air pollutants cannot go outside the valley, and therefore the pollution travels alongside the valley. This has led to the establishment of several infrastructure in the area metropolitana to monitor the air pollution. We have a system, Sistema Integrado de Alerta Temprana, which is called SIATA, which aims to analyze and monitor these air pollutants. However, this system has been focused on chemical pollutants, on the known aerosols. But what about the bioaerosols, which can account between 5 and 50% of the PM2.5? So that motivates my research work on my PhD, where I try to analyze and try to answer the question, how is the relationship between those meteorological conditions and climatological conditions, the air quality, and the composition of the aerobiome in an urban setting in Medellin? The aerobiome, it sounds very fancy, but it's just the airborne associated microbiome. We analyze this aerobiome in an urban area. For those who are familiar with Medellin, it's a monitoring station located near the Universidad de Medellin, which has several characteristics that were important for us. Mainly that the average pollution level it's not directly influenced by sources, meaning cars or industry, but the contribution of the emissions are mainly according of the wind transportation and the climatic regimes. To analyze this aerobiome, one of the main bottlenecks we had with the research related with the wet lab part was the DNA extractions, because the PM2.5, it's a very harsh environment for microorganisms to grow. So we developed and optimized a DNA extraction methodology to obtain enough DNA to be able to perform all the microbial ecology analysis. We started with some PM2.5 filters. This is what we're breeding. This is a sample taken in a period of 24 hours, a lot of smoke. Then we performed the optimized DNA extraction, then analyzed through 16S RNA PCR and nanopore sequencing, obtained the raw reads, and then coupled the Kraken, Bracken, and Silva database to perform the taxonomical classification. And we obtained the metadata, the climatic data from CIATA, which I mentioned before, and NASA Power, gathering some satellite data. Then we used several libraries to perform all the statistical analysis. I won't talk very much about this, and let's start with the main results. From the airborne in Medellin, at the film level, we saw that Protobacteria, Actinobacteria, Firmicutes, Cyanobacteria, and Bacteroidota are the main film that are present in the PM2.5. This was kind of expected. Some other studies performed in China, for instance, were able to identify this phyla within the PM2.5. When we analyzed at the genus level, we saw several genera present in the PM2.5. I won't read them all, but I would like to highlight it's that most of the most abundant genera present in PM2.5 were extremophile. And we also identify the presence of gut-associated bacteria present in the air. So we are also breathing fecal material. Surprising, isn't it? The core aerobiome, we define it as the genera present consistently identified in all the samples and which have a prevalence or a relative abundance, better, larger than 0.1%. These are the genera present consistently in the whole year we analyzed, but I would like to highlight what I said before. In the most abundant genera and in the core aerobiome, around 40 or 30, between 30 and 40%, it's extremophile. Regarding the environmental conditions, the climate season was the only one that actually had a disruption on the communities present, both in alpha and beta diversity. But what I would like to understand also was not only how is the structure of the community is changing in terms of the climate season, but also in the PM2.5 concentration. What we found was that the higher the concentration of PM2.5, it had a disruption in the microbial network, co-occurrence network present in the particulate matter. Finally, what I would like to highlight from this slide, we found some opportunistic pathogens differentially abundant on the PM2.5, particularly Weisella leconocost, are accountable for nosocomial infections. And what I would like to show you here are these two red arrows. We consistently found the presence of pathogens in the PM2.5, regardless of the climate condition, regardless of the PM2.5 concentration. So this is a red flag. We need to investigate and further deepen our understanding of the microbial communities present in the system of air quality monitoring systems. So concluding, this is the first study that analyzes the aerobiome present in PM2.5 using molecular techniques independent of culture. We successfully optimize a nucleic extraction, DNA extraction protocols, accounting for the low concentration and the large amount of inhibitors in the PM2.5. We identify a core aerobiome and the presence of pathogenic and also microorganisms that had the potential for degrading pollutants and aromatic compounds. The climatic season has a big impact in the overall structure of the microbial communities. And the concentration of the contaminants has the potential to disrupt the microbial co-occurrence network. The final message that I would like to give you is to keep in mind that the air quality monitor systems, which has been focused on complex chemicals mixtures, should also take into account the presence of potential microorganisms, not only for human health, but also for the environmental health. Thank you very much. I don't have, if you have any questions, I would be glad to answer it.
Great. Thank you, Simon, for your great and interesting talk. If someone have a question or a virtual one, let me know.
Just for the sake of curiosity, which would be the impact of breeding fecal bacteria on the human health?
It would depend mainly on the population of immunocompromised patients. So to understand what are the actual impact of breeding fecal material, more studies should be addressed to answer that question. But this is the first study, so it's a glimpse of the potential health risks due to the overall composition of the PM2.5. There is I don't know, I'm just letting you know what I think about from your question. There is an axis between the lung microbiome, the gut microbiome, and the brain. So it could have a direct influence in the overall health, particularly in inflammatory diseases. But more research has to be conducted to correctly answer your question. Thank you. Any other questions? Okay, thank you. Over here. Well, I think something that might help, it would depend mostly what areas are you being in the city. There are some areas that are more polluted than others. Also, we have a larger amount of gas exchange. We are breathing all the time. This filter is 24 hours, filtering all the time, but there isn't a flow outwards the sampling device. So we are in the breathing process, we have some volume intake, and then you breathe out, and you're also expelling some of these particulate material. But the filter that I show you, it's for 24 hours, filtering the air and larger volumes of air, larger than our lungs. Okay, thank you.